one of the privileges of writing software is that you get to learn things by osmosis. You get to collaborate with people who know everything about something. And so one of the privileges for me in my career has been my collaborations. The next two talks are going to be, so one of them is going to be quite short. Nancy Kanwisher's here. And Nancy is someone who's both a collaborator and a friend of mine. And it's very nice of her to pop in. So she is someone who's taught me a huge amount about brain function and functional imaging and kind of how we do science in general. And then after Nancy, David Slot's going to give a talk. David came as a postdoc when we were kind of messing around in morphometry. We kind of knew we could measure cortical thickness, but we had no idea what to do with it. And David brought a level of scientific rigor that really pushed us to make things work to study human brain morphometry. And so these will be the next two talks. So Nancy's up first, I think.
Hi, everyone. So I'm just going to take a couple of minutes to do what Bruce told me I couldn't do, which is express great appreciation, because almost nobody, possibly nobody, has been as important as a collaborator, as a kind of scientific consultant and a friend, and just a really important person in my career than Bruce. So a huge thanks for all of that. And everything I've done pretty much in my the last 20 years of research in my lab has used Freesurfer. So much so that when I tried to think of what is the difference that Freesurfer has made, I don't have the control condition. I've never done anything else. So it's been like absolutely fundamental, but in all kinds of ways. And so I just thought I'd mention two things that to me I think are really special and important about Freesurfer. And so one is just that, as I'm sure all of you guys know, Freesurfer it makes it very transparent and easy to interface and write your own code and interface with the tools in Freesurfer. And I think that's really important in our field, because it's only when you really kind of mess with your own data that you know what the hell is going on. And to me, there's like when I look at the work in the field, there's a real correlation between the people who use Freesurfer and the quality of the science. Some of the other packages that shall remain nameless are really kind of associated with, that are sort of more black box, shove your data in and get a paper out the other end, are sort of really have led to much weaker science, I think. And so that's one thing I really love about it, is how I can tell anybody in my lab, like, learn Freesurfer and then write all this code to use it and interface with it and do your own thing and, control your own data. But the other thing is the spectacular visualization tools. And as I was thinking about this, thinking, why is that so important? It's not just that we all like pretty pictures and it helps us, get people to pay attention to our stuff and to, publish our stuff in higher profile journals. It's also that that visualization is actually how we understand data. And as I was thinking about it, the reason is that we all have this spectacular computer back here in visual cortex, right? Half the cortex. And we can use that computer to understand our data if we visualize the data in a way that we can feed it in and use this to understand it. So it's really an understanding tool, not just a presentation tool. And I think there's nothing as good as Freesurfer for doing that beautifully. So in my lab, just briefly, we've used Freesurfer's fabulous tools to do all kinds of stuff, from mapping out some of the regions in the ventral visual pathway to looking at how the organization of retinotopic cortex changes in people who lose foveal vision due to macular degeneration. These are all things where you need to be able to see the cortex laid out in a really nice way. To looking at what we think are homologies between humans and macaques, again, by laying out the activation patterns on the surface of the brain and seeing, for example, that this selectivity that we and others have seen for a long time on the bottom of the brain for going from medial to lateral, it goes faces, color, places, lateral to medial, on the bottom surface of the brain, and it goes in the exact same order on the lateral surface in the monkey brain. And that's just such a weird coincidence that just looking at that, to me, suggests that those whole systems are probably homologous, like the whole thing probably just rolled over around the bottom surface of the brain. Yeah, just to say, it's been fabulous for me. It has like made my whole career possible. I am hugely grateful, and thank you.